Okay, so as in the last video, we have discussed that how urine is concentrated or diluted in the kidney. Alright, so today we are going with the topic which is counter current mechanism. Alright, so I should write here the meaning of the counter current first so that it can be clear to you. So, wait, let me write here the topic first. Alright, so today we will be discussing about counter current. Alright, counter current. Now let's see what's the meaning of counter first. Alright, counter means opposite. Alright, let me write here down. Counter means what? It means opposite. Opposite. And current means here in the biology, wait, the movement of a liquid. Alright, it is what? It is the movement of the liquid. Movement of the liquid. So as the name suggests, it is the opposite, like it is the movement of liquid in the opposite direction. Alright, because counter means opposite and current means the movement of the liquid. Alright, so it is suggesting what? It is suggesting that it is the movement of liquid, that too in the opposite direction. So let me write here the definition. Alright, okay, so it is what? It is the movement of liquid in opposite direction. All right, in opposite direction. So it is the movement of the liquid in opposite direction. All right, so for showing you guys that the movement of the liquid in opposite direction, I have to make here the diagram of nephron first so that I can show you how the movement of liquid in opposite direction is happening. All right, so let me make here the nephron first so that I can explain you with that. All right, so here we have the nephron which I have made so many times. All right, here I am making the PCT. This is what? This is PCT. Here we have loop of Henle, which is descending loop of Henle. All right, here we have ascending loop of Henle, and here ultimately what do we have? We have DCT, and here we have collecting duct. All right, so here I have made the diagram, so let me label it first. All right, this is Bowman's capsule, Bowman's capsule. This is PCT. Wait. All right, so this is DCT we do have, and here ultimately we do have collecting duct. Collecting duct. All right, so here I have labeled this diagram, as you can see. So here we will have our filtrate, and in the filtrate the like what will be happening, like there will be osmolarity of 300 osmol per liter. All right, we know that, we have already discussed that. And here in the PCT that would be 300 again, And as I told you, the descending loop of Henle is permeable to the water, but it is impermeable to the ions. So here the whole water would be gone out. Alright, this water would be gone out. This H2O would be gone out, again gone out. Alright, this would be gone out, this would be gone out, and here we will have the highest concentration of ions here in the, all right, because we know that in the medulla the concentration of ions is getting greater and greater. So let me make here that as well. In the PCT, like PCT, Bowman's capsule, DCT and collecting duct are where they are in the cortex of the kidney. But talking about loop of Henle, including descending and ascending, they both are in the medullary region. Alright, so this is what? This is medulla. This is medulla. Alright, so here the concentration would be very much larger. Alright, so here for suppose it is 1200. Then again we know that the ascending loop of Henle is permeable to the ions, but it is impermeable to the water. So here we'll have pumps, and those pumps are known as sodium potassium pumps. These are known as sodium potassium pumps. Sodium potassium pumps. Alright, so from here the sodium will be gone out and chlorine would be like in. Alright, here the ions will be going out, because we know that so water cannot go out from here, just because this layer is impermeable to the water but permeable to the ions. So here we will have, wait, sodium ions which will be gone out. Alright, and this is the direction of urine. So here as you can see, what's the direction of the water? Wait, I'll tell you that later. Let me complete this first. Alright, so here some urea will be reabsorbed too. Alright, here some urea would be reabsorbed too from this DCT. Alright, and ultimately here we'll have our urea again here, and here we will have our urine. Alright, so this is the diagram of nephron which I have made so many times. Again I have made for you guys. Alright, now as you can see in the loop of Henle, as well as in ascending and in descending too. Alright, so let's see in the descending first. Like see, the descending loop of Henle, in the descending loop of Henle, how the urine is going. We cannot call it urine, because urine will be called when it will pass through the collecting duct. In the collecting duct urine will be formed, but here we have to say filtrate. Alright, so see the direction of the filtrate, how it is going. It is going like this. Alright, as you can see the direction, it is going down. Again see in the ascending loop of Henle, here what is the direction of the filtrate? Alright, let's see. It is like this. See, this is going up. From the descending loop of Henle it was going down, as you can see here. This is for descending loop of Henle, and this is for ascending loop of Henle. Alright, so as you can see there, the directions are completely changed. They are in opposite direction. The one is coming down and another is going up, and this is the movement of liquid. Alright, so the movement of liquid, what we studied in the countercurrent mechanism, it is the movement of liquid in opposite direction. So as you can see here, the filtrate is moving from up to the down, and again from down to the up. So the direction of liquid is in opposite direction. So this is what we call the counter current mechanism. Alright, so here let me write some important points. Alright, I am using white color for that. So first point, urea cycle is in liver. Alright, I am telling you now the key points. Now the key points I'm telling. Urea cycle is where? It is in the liver, as we have seen. How this is in liver? Because as I told you in my previous lecture as well, we were having proteins, all right, and those proteins were break down into the amino acid, because we know that protein are made up of the chain of amino acid, and then that amino acid was going to this liver, all right. In the liver we know that the formula of urea, not the urea but the amino acid, C was here in the middle, then again here's carboxylic group was here, it was acidic group, here we were having amine group which was NH2, all right. Here we were having hydrogen, and here we were having radical, all right. So we were cutting this, we were doing deamination, and here it was combining with what? It was combining with carbon dioxide, and it was going to the cycle which was urea cycle, all right. So urea cycle is where? It is in the, it is in the what? It is in the liver. So don't confuse with that. If someone asks you where is urea cycle, so you don't have to say urea cycle is in nephron or it is in kidney. No, not at all. It is where? It is in the liver, all right. It is in the liver. So let me tell you another thing. Wait, let me do this first. My things are removed actually. All right, I'll make it right. No worries. This is urea, right? Urea cycle. This is cycle in liver. All right. So second most important point, urea recycling is in kidney. If someone is asking you where is urea recycling, so urea recycling is not in liver but it is in kidney. All right. Urea recycling. Recycling. And it is where? It is in the kidney. It is in the kidney, all right. So third point, sodium and chlorine and urea are main component for the concentration of urine. If you want to concentrate urine, so what are the main components of the urine? Sodium, chlorine ion, NaCl. Wait, let me write here down with this color. So third point, what do we have? Sodium ions, chlorine ions, and urea. Urea are the main components for urine concentration. Components for urine concentration. All right, concentration. Okay, so again we do have important point. What that is? Medulla is highly concentrated just because of sodium, chlorine, and urine. Because we know that sodium, chlorine, and urea are present where, as they are the important components for the concentration of the urine, and they are present in the medulla. All right, they are present where? They are present in the medulla. So let me write this point as well. Medulla is highly concentrated just because of the sodium ions, chlorine ions, and urea. Concentrated. All right. So the end of the collecting duct is permeable to urea and ascending limb too. All right. So this ascending limb is what? It is permeable to the urea, as well as this collecting duct is permeable to the urea as well. All right. So let me write this here. What I said. End of collecting duct is permeable to urea and ascending limb as well. Alright, so not whole collecting duct is permeable to the urea, but as you can see, only the end of the collecting duct from here. Let me mark this down. Only this part is permeable to the urea, and whole ascending loop of Henle is permeable to the urea. Alright, so this was all for today, as I told you about the counter current mechanism. So in the next video we would be going with the um, counter current exchange. Alright, that is very important topic, because to save ions in the medulla we do have a lot of ions, but they are all going uh, out, like, alright, so we have to save those ions. So to save those ions we do have vasa recta. Now in the counter current exchange, vasa recta has to play a role. Alright, very important role vasa recta will be playing. So vasa recta will save those all ions, and the saving of those all ions is what we call the counter current exchange. So in the next video we would be going with the counter current exchange, and this was all for today.